Translation begins when a ribosome attaches to an mRNA molecule in the cytoplasm. Translation initiates at AUG. Remember, AUG is the start codon, and it codes for methionine. tRNA molecules carry the amino acids to the mRNA. The tRNA molecules have anticodons on one side and amino acids on the other side. This fact is really important because the anticodon on the tRNA matches up to the codon on the mRNA, and whichever amino acid is attached to that tRNA on the other side is the amino acid that gets added to the polypeptide chain. So each anticodon, each type of tRNA, one anticodon has one type of amino acid, and that's determined by us based on the genetic map that we just saw, the genetic code table. Since AUG always codes for methionine, the first amino acid brought in for every round of translation is methionine. As each codon passes through the ribosome, more tRNAs bring the proper amino acids into the ribosome. And this is determined by whichever tRNA has the complementary anticodon to the codons that are in order on the mRNA. Each transfer RNA has an anticodon whose bases are complementary to the bases of a codon on the mRNA strands. The tRNA attaches its anticodon to the appropriate codon of the mRNA. There's the anticodon and the tRNA. We see that the anticodon for methionine is UAC. So AUG is the codon. A always has a complementary base of U. A and U form base pairs. C and G form base pairs. So if the codon is AUG, then the anticodon must be UAC. And every tRNA that has UAC as its anticodon is going to have methionine attached to the other side of it. So if we look at some of these other tRNAs, we see that the mRNA codon UUC attracts the tRNA anticodon AAG, and the tRNA anticodon that has AAG has a phenylalanine attached to the other side. So the methionine is going to be attached to the phenylalanine, and then that's going to be attached to the lysine, and so we are starting to develop a polypeptide chain based on the order of the codons, because the order of the codons determines which tRNAs are going to be attached in which order, and then the types of tRNA determine which amino acids are going to be added to the polypeptide chain. The ribosome is the blue structure the blue and gold structure that we see here that's actually making the connections between it's actually reading the mRNA and making the connections between the amino acids. So here we see a connection between the methionine and phenylalanine. At the same time, the bond holding the first tRNA molecule to the amino acid is broken. This is a weak hydrogen bond, and it's easy for the ribosome to break it off and send the tRNA back to pick up a new piece of methionine. The tRNA moves into a third binding site from which it exits the ribosome. The ribosome then moves to the third codon where tRNA brings in the amino acid specified by the third codon. In this case, we don't see the names of the bases indicated, but we can tell by the color coding that the next codon is going to be CU G. So that means that the anticodon must have GAC. The product of transcription, let's get these terms clear. The product of, actually, let's start with replication. The product of replication, we start with one DNA. The product of replication is two identical DNAs. After replication, we have transcription, which takes place in the nucleus. The product of transcription, we start with one DNA. Our product is mRNA. That mRNA then moves to the cytoplasm and goes through translation. The product of translation is a protein made from the polypeptide chain that's made according to the instructions in the mRNA. The amino acids that make up the protein are picked up throughout the cell by the tRNA molecules and then carried over to the ribosome to be combined into what we see as becoming a long polypeptide chain. The translation process continues until the ribosome reaches one of the three stop codons. The stop codon in this illustration is UGA. So when the mRNA, when the UGA codon reaches the ribosome is going to stop making a polypeptide chain, and the existing polypeptide chain is going to break off. Different parts of the polypeptide are going to be attracted to each other, and so the protein will fold over, or the polypeptide will fold over in a predictable shape, and that is what we call a protein. A protein is often made of a great number of amino acids that fold over in a predictable way, so the shape of the protein can be predictable. If the stop codon had mistakenly been made into a regular codon, then the translation would not stop and another amino acid would be attached, and the protein would have an error in its structure. It would be flawed. Our polypeptide chain should be complete at this point. Our mRNA will separate from the ribosome, and the polypeptide chain will also separate and form a protein.